Thank you everybody for coming. And let's talk about AlphaFold. So I'm talking about structure prediction using AlphaFold. And very briefly at the end, I'm going to be talking about design. There's already a lot to talk about with how AlphaFold works, how to run your predictions, and how to interpret it. And then there's a little bit at the end then about how to design your own proteins. So this is the overview of the talk. I'll talk about AlphaFold under the hood and how to interpret it, how to run your own predictions, some tips, and then some applications for structure prediction. And so why might we want to predict a structure of a protein? Well, first of all, it's nice to know what your protein looks like. It's nice to give structural context to a biological question. It's nice to create these initial models for cryo-EM and crystallography. If you're trying to build a model, you have a map and you want to put something in that you can then refine, right? This is very useful. It has been very useful to run a prediction. It's useful for guiding protein construct design, and for a lot of biochemistry projects, it's useful to have an AlphaFold model to then use when you're designing constructs. And really what you want to do is generate testable hypotheses that you can then use other methods to test if these hypotheses are correct or not. And so this progress towards predicting a protein structure purely from its sequence was this global project that was managed by CASP, where algorithms would be submitted to CASP and they would be tested against proteins they had not seen before, sequences that they had not seen before, and then the performance was dictated by this global distance test. And you can see AlphaFold came out in 2018, did really well, and then AlphaFold 2 had a really high global distance test metric. And from that point on, AlphaFold has become part of our workflow as biochemists or structural biologists. And you can see here the performance of AlphaFold 2 was very good. Now we have, of course, AlphaFold 3. I mean, since then, we've had AlphaFold Multimer, which deals with multimeric proteins and multiple proteins, not just one. And then now we have AlphaFold 3, which also allows you to use different kinds of biomolecules, RNA, DNA. And really, when we say AlphaFold, we're referring, it's an umbrella term for all three of these things. So running an AlphaFold prediction is essentially encompassing these three things, and we'll go through some of the differences and what they mean. But essentially, in order for them to be able to do this, they had to train neural networks in order to turn a sequence into a structure. And so what we need in order to train a neural network is to have examples of sequence structure relationships. And this is what's available already on the protein data bank. A lot of people put a lot of work in to solve structures of proteins. And so we know that a given sequence should look like this, for example. And so this can then be used to train a model to then, for sequences it has not seen before, to generate a structure. But another really important insight was that rather than using the sequence itself, one can use a multiple sequence alignment. So here's, you have this sequence that we're interested in, and you can see in the sequence alignment, we can see that these two residues are co-evolving. They seem to be following similar evolutionary paths. And so one can interpret this as meaning that these residues are probably close together in space. And so you'll notice that the input to AlphaFold really is a multiple sequence alignment, and that's the way you should frame it in your mind. It's the multiple sequence alignment is what goes in, and that's what ends up giving you the prediction, because there's a lot of information that's held in here. And so these are the multiple sequence alignment structure predictors. So this includes AlphaFold and RosettaFold, which came from David Baker's lab at the Institute for Protein Design. Again, these are generating multiple sequence alignments that go into these trained neural networks that end up with a structure. But we also have these protein language model structure predictors that you should be aware of. And these do not take multiple sequence alignment. Rather, they have the evolutionary relationships embedded within them. And so you don't essentially have to generate those evolutionary relationships. They're already within them. And this ends up being much faster, but it's also slightly less accurate. So it's good for running whole genomes. People have run whole genomes with, say, ESM folds from Meta. But if you want a more accurate prediction, typically you would resort to generating or curating your own multiple sequence alignment. Okay, so how does it work? So for, like I said, well, you have a sequence, you want to generate a multiple sequence alignment. Now what happens is that this goes into their trained neural network. First, you have this MSA module that turns this multiple sequence alignment into an initial set of XYZ coordinates. So it infers from these relationships where these residues should be in space. And then these initial coordinates are turned into something that looks like a protein when it goes through this structure module. And so for AlphaFold 3, this general workflow did not change. So we'll have a multiple sequence alignment that's turning into some initial coordinates. But instead of a structure module, now we have a diffusion module, so a generative model. I wouldn't worry about it. Essentially, it's a different way of turning the initial coordinates into a structure. And because it's different, the results can also be different. And I'll talk later about the differences between AlphaFold 2 and AlphaFold 3. But essentially, this is the workflow. And something else you can, in theory, put into this model is templates. So you have a sequence, and this little alpha helix is found in other sequences that already have structures. You can think about injecting that known structure into your pipeline. This is what we call templates. But it turns out that that's not as important. The multiple sequence alignment actually holds most of the information you need in order to generate an accurate prediction. It's just good to know that these are potentially part of the pipeline. What is really important, however, is this recycles. So when you get your first structure, it's actually not very good. But it turns out what's really important is recycling it back to the beginning, and then you end up with iteratively better and better structures. And so this recycling is something that's important to think about, and we'll talk about how you, as a user, have to think about the recycling. But essentially, eventually you end up with something that's really nice. Now, of course, you're only as good as your multiple sequence alignment. And the way that this is generated is by searching through a sequence database. So with AlphaFold, originally, this was searched through UniRef90, Magnify, and BFD. So these are large databases from different organisms. And this has been maybe a limiting step for local implementations. Actually, this sequence database search is slow, I would say. And so you may have heard of ColabFold, which is a Colab implementation of AlphaFold that I'll talk about later. And what's nice about this is that it uses a clustered version of these databases. So it's actually really fast, and it's using this MMCX2, and I'll talk about later how one would run ColabFold. Really, there's a lot more going on under the hood, but as a user, this is probably the level that you need to understand it in order to do your work properly. Because really all you need to give this thing is your sequence. And then what you get out at the end is structures, right? So with AlphaFold, you get, AlphaFold 2 or AlphaFold 3, you get 5 structures. And these are generated by models that AlphaFold has trained slightly differently, so slightly different inputs. And basically, this is 5 shots at getting the correct structure. You can think about it that way. Then you have your multiple sequence alignment metric. So this is, again, you generate multiple sequence alignment, and one could interpret how well that went, how many sequences were pulled. And then you have these confidence metrics. So these are telling you how confident you should be in this prediction. You can ask a question of how confident am I in this residue specifically, or you can ask about how confident am I in residues relative to one another. So we have this predicted local distance difference test, which is per residue, and this predicted aligned error, which is pairwise. So I think these are really important. So we're going to go through them so you can correctly interpret them. And so for this, I'm going to use an example of something I work on. So I work on the Dynein motor complex. So this guy over here has a motor domain, and it walks along these microtubules. It's part of this enormous complex. It's got dynactin. It's got this called coil adapters. And part of the Dynein complex is this Dynein light intermediate chain. So it has this globular domain, and it's have these little alpha helices on loops. So it's kind of a nice example for this. And you can see that when you predict just this light intermediate chain in isolation, AlphaFold gives you these 5 structures. You can see it's got this globular domain, and it's placed these alpha helices kind of randomly in space. And there's a question of how am I supposed to interpret this result, right? First of all, these 5 structures that it gives you are already ranked when you run an AlphaFold prediction. And this ranking over the years has changed in terms of how it determines how one structure is better than the other. Historically, it's been either average PLDT. We'll talk about the PLDT. There's a predicted template modeling score, some combination. In practice, really, you want to be looking at all of them, and really you want to be interpreting the PAE plot typically if you're looking at multimers. So we're going to look at these confidence metrics for one of them, and then you can basically apply that to the other ones. So the first output from, let's say, ColabFold is your sequence alignment, right? So your sequence gave you this multiple sequence alignment after running it through a database. And here, each line is representing a stretch of sequence that was pulled from that database. And we have here a concept of MSA depth. So here in the middle region, we have a lot of sequences. So there's a high MSA depth. And on the edges, we don't have that many sequences. So there's a low MSA depth. One can interpret this as meaning that you're going to get more confident predictions of the center of this protein than you are of the edges of the protein. And really, a rule of thumb here is that you want at least 30 sequences, which, from this example, you can see shouldn't be that hard. But for divergent organisms, this MSA depth can be kind of challenging, right, for getting even more than 30 sequences sometimes can be really hard. So the first confidence metric that I want to talk about is the predicted local distance difference test. So this PLDDT. And again, this is a per residue analysis. And what this is really good for is understanding the secondary structure of the prediction, right? So if something has a high PLDDT, it's probably a structured residue. So it's good for secondary structure. And if something has a low PLDDT, then it's good for disorder. The general rule of thumb here is that if something is less than 50%, it's probably disordered. Something is higher than 50% is probably ordered. I mean, it's a bit hand wavy. This is their guidelines here. You know, we have high quality, probably correct. But essentially, you can see with this example that I'm using, these loops are in these low PLDDT regions. So these are probably disordered, so randomly placed in space as well. And then we have this ordered alpha helix, right? We have secondary structure here, and this is coming out as higher PLDDT. So if this is something that you're interested in interpreting about your structure, whether or not the secondary structure is correct or it might be disordered, the PLDDT is a very good metric for that. A little bit less intuitive is the predicted aligned error. So this is an interpretation of how confident are you in the position of one residue relative to another residue. And for a lot of what we work on with protein-protein interactions and domain interactions, this is what we care about. We care about knowing whether or not these are confidently predicted. So you get this 2D plot, which is one residue versus another. And let's look at one. It's a little bit counterintuitive because what they're plotting here is what is the expected error of residue 1 if you align the prediction and a true structure at residue 2? But we really don't have a true structure. This is something that they've trained a model to interpret the prediction to try to understand what the confidence is. So they already had these examples of sequence to structure relationships. And so they can train a model to learn how confident they are in those predictions. And then when they run their predictions through that confidence model, and then they can get these PAE plots. And you can use this to interpret your structures. And it's actually a really powerful part of AlphaFold, the fact that you have these confidence metrics. And so let's look at it more closely. So we have a trivial example. What is the confidence of residue 198 relative to itself? Obviously, it's very confident. So we have a very low error. So just be careful with the terminology here. A PAE, so a low PAE is a low error, which means you're very confident, right? I see people make this mistake a lot. But so that's why we have this diagonal over here. So every residue is confident with itself. That's the reason for this diagonal. We take a more interesting example. We look at this part of the plot. That's this residue, 198 versus 113. I mean, arguably not an interesting example, but I'm just using it here to show that, I mean, these are not related to each other in space. And that's coming out as something with high error, right? So very low confidence for the position of these two residues relative to each other. And this is, in theory, a symmetric plot. It's not really, I'll talk about that later, but one really only needs to worry about one triangle here. So you can start interpreting, what residue is correct relative to another residue or confident relative to another residue here. So if we look at subdomain, for example, right? So we have here a little patch. And what's happening is that not only is the residue confident with itself, but it's also with its neighbor and its immediate neighbor and its other neighbor and neighbor and neighbor. And so then you get this stretching away from the diagonal that is essentially what a subdomain would look like in this PAE plot, right? So whenever you have a patch, that is an ordered part of your prediction. If we keep going down the sequence, we have this part that is disordered, right? So there's nothing really to say here. And then we have this other domain or subdomain where you have this patch. And then this subdomain is interacting with this subdomain. And so you end up with a patch that's away from the diagonal, right? So we have different parts of the protein, different parts that are interacting with each other, right? So residues that are far apart that interact with each other. So when you're interpreting the PAE plot, essentially what you're looking for are these patches, ideally away from the diagonal. Now, it's not so useful for things like, alpha helices. Like here is this alpha helix. In the PAE plot, right, you're only confident with respect to your immediate neighbor. So this doesn't really come out in the PAE plot as clearly. So if this is something that you're trying to interpret from the prediction, you would rather look at the PLDDT, which is giving you more per residue confidence. By the way, stop me at any point. I think it's useful to ask questions during rather than at the end when you forget. All righty. So the next thing is multimers. So what's happening with multimers? But really, it's the same thing. Instead of giving it one sequence, now you're giving it two sequences. And this is going through, if you're using AlphaFold 2, now it's going through AlphaFold 2 Multimer. If you're using AlphaFold 3, it just handles it. And so for all the implementations that we have, especially at the LMB, AlphaFold Multimer is used by default when you give it multiple chains. So really, you don't have to think about it. You're either using AlphaFold 2 or you're using AlphaFold 3, and you're going to get these outputs anyway. And with multimers, the outputs are the same. You have a PLDDT, and the PAE plot is really what's important for multimeric predictions, because typically what you're interested in is this protein interacting with this protein? So what you're asking about is the PAE low in regions where the proteins are interacting? So let's look at one more closely. Here's this PAE. I've just split it up into 4 sections. And these top left and bottom right sections are essentially the same as what I showed you before. It's the PAE of just protein A, right? So you have this diagonal, and you have things that are away from the diagonal representing subdomains, and you have these red bits that are the loops. Same thing for protein B, right? We have this diagonal and these domains. But really what we're wondering is what is the confidence of the interactions between these two proteins? So we're looking at these quadrants that are away from the diagonal, so the bottom left and the top right. And it can be quite hard to interpret these things. So I find things like Chimera useful, where you can load up your AlphaFold prediction and the error plot, and then you get this funny color thing where you can drag a little square, and then it will show you where in the PAE plot you are in your structure, which I find very useful. So for example, if I've dragged this, if I highlighted this region here, you can see I've highlighted essentially all of protein B. So all of it is highlighted, and just a little bit of protein A, that's this little loop. And because the PAE here is of poor confidence, right? So it's high error. Then you can see that obviously these are not interacting or not confidently predicted relative to each other. So that makes sense. Another useful thing is this web app. It's called PAEViewer. So this is something where you can load up your prediction again, and not only do you see the same thing, but you can also highlight your sequence. It highlights your sequence for you, and you can load up these cross-linking mass spec results onto it, and it will show you where the cross-linking results are in the PAE plot, which is kind of handy. Okay, so there's this question about whether or not the PAE is always symmetric. I get this every year. And here's just a nice example of where it is not. So we have protein A interacting with the slow peptide protein B. And you can see protein A is very nicely globular. Everything is interacting with everything very confidently. And then in protein B, we have also this peptide. You have like basically what looks like an alpha helix. And then what we're interpreting is whether or not we're confident in the interaction between protein A and protein B, which is over here and over here. And it's strange because over here, the error in B is low when we're aligned at A, but here the error in A is high when we're aligned at B. So it's kind of weird. Why would the error be different if you're aligning one versus the other? And it turns out, it's not very intuitive, but it turns out that this asymmetry occurs when the local conformation of one of them is very uncertain relative to the other one. So if you're aligning to something where the local conformation is very certain, then the alignment of the other one will not change very much. But if you're not very certain about this guy's local conformation, then the alignment of the other will be all over the place. And so you end up with this really high error. I wouldn't worry about it. Just take it as a warning flag. When you see something like this that's asymmetric, probably one should be a little bit skeptical about the prediction. Doesn't mean it's wrong, doesn't mean it's right, but just something to watch out for. Those are the outputs. We have the 5 structures, we have a multiple sequence alignment and these metrics. And if you're using this prediction for a manuscript, let's say, you wouldn't typically put in the multiple sequence alignment. You might mention it, let's say. If you're interpreting secondary structure, you would probably either put this PLDDT plot, or you would map the PLDDT onto the structure using something like Chimera. If you're interpreting interactions, I would say you have to submit the predicted aligned error. Otherwise, how would one know if the prediction was confident or not? So the PAE plot I find very important for submitting manuscript when you're submitting your manuscripts. And the model itself you can actually submit into something like Model Archive. So if you have a prediction that was very useful for interpreting a protein interaction that you then use to make mutations, it's very useful to put it somewhere so that somebody can take a look, download it for themselves and take a look and not have to run it themselves. And so something like Model Archive is nice. Here's just a recent example from Kelly from our lab just in March, where George had deposited this structure of his from AlphaFold 3. So it's nice because then you can just go in and pull it out. Okay, so that's how AlphaFold works, how to interpret it. Are there any questions on the first bit? So let's talk about how you can run your predictions. The easiest way to run a prediction is to just fetch one that has already been run for you. So there's databases like the AlphaFold protein structure database, where you can just put in your protein and it will give you the prediction for that protein. This is actually embedded into the Uniprot. A lot of people use the Uniprot, and there's a section called structure where you have experimentally derived structures, and then also typically you'll have the AlphaFold structure for that protein. So here's just a random one I pulled out, RL3. You can see it's there's an AlphaFold structure that you click, and then it shows up, and it shows you the PLDDT that's colored on top of it. One should be aware, though, that there are experimentally derived structures in this list that might be more useful for what you're trying to interpret. So make sure you look at those rather than the AlphaFold structure, the prediction, sorry, because they might be, they might hold the information that you want. Also be aware that if you're looking at a protein, probably a homologous protein to the one that you're looking at has an experimentally derived structure, right? So let's say the mouse version of the protein you're interested in might have a crystal structure, and then you might want to interpret that rather than an AlphaFold prediction of the protein that you're interested in. It's also not very useful for things like obligate dimers or things that don't exist in isolation. For example, this is BICDR. It's a coiled-coil protein, so it's a dimer. And naively, you would say, oh, this is just an alpha helix, right? A long alpha helix. But really, we know that this is a coiled-coil protein. And for that, really, you want to be running your own predictions. The easiest way to run your own predictions is to use these Google Colab notebooks. So if you go to this website, you'll see that there's a list of them essentially for different versions of these different algorithms. There's this AlphaFold 2 mmseeks2. This is what I've called ColabFold, or what they have called ColabFold. The reason it's called ColabFold is because they're on these Google Colab notebooks. And so these are hosted by Google, and you can use Google's GPUs to run your jobs essentially. And it's very simple. You put your sequence and you wait, and it will run on their GPUs, and you get your output. There's a bit of a limit to this. So you have to leave the page open. Otherwise, it times out and it gets annoying, and you can't run really big things. I think 2000 is the maximum length here. But for quick and dirty things, this is actually very useful. Now, at the LMB, we're lucky that we have this really nice GPU cluster where we can submit our jobs and essentially walk away from it and have the nice GPUs of the cluster handle it. So this is handled by scientific computing. So I urge you to go to this website and check how to register to the cluster if you're interested in doing this, what the fair usage is. And eventually, once you have that set up, you can add these to your path. If you don't know what this means, just feel free to send me an email. And then once that's set up, all you need is your proteins that you want to predict. For example, I have a complex here of protein 1 and protein 2 in a FASTA format. So there's a header and a sequence, a header and a sequence. And then you just submit the job from one of the supercomputers, say ColabFold, and then your complex.fasta. And there's a lot of options that you can play with. If you just run ColabFold without any arguments, then you'll see the list of options that you can play with. But essentially, when you submit this to the cluster, what's happening in the background is that these sequences are sent to a server for generating the multiple sequence alignment, right? That's the first step. And then it goes through an AlphaFold 2 prediction, and then you end up with the results into the folder that you've specified. And then just keep in mind that these are communal services. So the mmseeks online server is a communal service, and also the cluster is a communal service. So just be mindful when you're submitting a lot of jobs that there's other people that are also using these things. But the parameters that you can play with here are, for example, the number of recycles. I mentioned earlier, recycling is really important, right? So the more recycles you have, the better, up to, let's say, 20 recycles. I mean, here's an example where, this protein didn't really plateau in quality until about 12 recycles. Right now, it's handled such that it will continue recycling until it hits some kind of tolerance, or it hits 20, whichever comes first. So you really don't have to worry about it anymore. The other thing that you can change is a custom template. You can provide it a custom template. So something that it can use as a starting point to then predict. In practice, I haven't had this work very well. I know other people have also struggled with getting custom templates to work, but I'd be happy to hear if somebody has had any luck with this. Something else that you can do, the thing about AlphaFold is that it will predict, for example, the closed state of a protein, wherein that there's a closed and open conformation, but it can just continue to predict the closed conformation. If you want to kind of help it out of that local minimum, you can, for example, reduce the MSA depth, right? So you reduce the number of sequences in the multiple sequence alignment, and essentially what you're doing is introducing uncertainty, right? The less sequences you have in your MSA, the more uncertain the model will be about the structure. And that's a way of helping it get out of those local minima. And another thing you can do then is increase the number of seeds. So this increases the number of models. So there's a slightly different input to each seed. I wouldn't worry about it so much, but the combination of increasing the number of seeds and using dropout apparently has been something that people have found helpful when they're trying to get these different conformations. If you're trying to build models, so let's say you have a cryo-EM density and you want to run a prediction so that you can then refine it into your cryo-EM map, typically what you would do is run an amber relax. So this is to help get the side chains to be of reasonable geometry, right? So what AlphaFold will give you is sometimes a bit mangled and the side chains might clash with each other. And so just running it through an amber relaxation step is useful because then the side chains make a little bit more sense. And you can use that as a proper input to your cryo-EM model building. Okay, so that's AlphaFold 2. It's nice, customizable, and we can run it on the cluster. Now with AlphaFold 3, it's a little bit less customizable, but it's on an online server, which is nice. So you essentially go to this website and you can run your AlphaFold 3 job. So this is a protein sequence that one could submit. And what's really powerful about AlphaFold 3 is that now we have the inclusion of other biomolecules, right? So you can choose, for example, a ligand and say, include an ADP molecule. So each one of these will have a drop-down menu, and you can, say where, predict where the ADP would also go in this protein structure. Doesn't mean it's going to predict the ADP state of that structure. That's not always going to be true, but sometimes it's true. Sometimes, putting an ADP in there will get it to predict, some ADP conformation of a protein. In that sense, it's quite simple. And here's what do I want to say here? So yeah, there's also another something that you should just be aware of is that RosettaFold All Atom is also another software that you could use to predict different biomolecules. So it's not as nicely in a web server, but it's also very powerful. This comes from David Baker's lab. So if you're interested in that kind of thing, you can check it out. But so here's an AlphaFold 3 result of a very large protein. This is a kinesin. And you can see that the web server will give you this nice display. It's got the protein. It's colored onto it, the PLDDT. It's also got the PAE. PAE is not as nicely segmented, so it's not clear really where you are in this sequence. But you can always download this result, put it through the things that I already described, and then start interpreting your structure. And you can see, this kind of thing would take very long on AlphaFold 2, even in our cluster. And what's nice about AlphaFold 3 is that because it's on their servers, it's very optimized. They already have everything set up really nicely. And so things happen really quickly. So it's very nice for very big things. Not very customizable though, so you can only really play with the number of seeds, which I mentioned earlier. So you can increase the number of seeds, and people have found that having a lot of seeds, so basically a lot of models can therefore get you the variation between the models that you're looking for if you're looking to find different conformations. But essentially, that's the only thing you can play with. And really, the annoying thing is that you can only run 30 jobs on the server, although very useful to have this resource. And eventually, people are coming up with ways of getting this locally so that we can run it on the cluster. There's some examples of this now, like bolts, but it hasn't been as trivial to set up at the LMB yet, but we're working on it. Okay, so here's just a difference between AlphaFold 3 and AlphaFold 2 that I could come up with quickly. AlphaFold 3 obviously is super powerful because it handles different types of molecules, whereas AlphaFold 2 can't handle DNA and RNA. And it's super fast, accepts up to 5,000 amino acids, which would be, for 5,000, that would be very difficult for AlphaFold 2, even on nice GPUs. The annoying thing is that it has limits. And what's really important for us is that it produces more hallucinations. At least that's what's been the experience of people at the LMB. Because of this diffusion module that's used at the second step of the prediction, turns out that it creates more alpha helices where there would otherwise be disorder, for example. So one needs to be careful. If you have something small, I would rather use AlphaFold 2 that you can submit to the cluster. Although it only handles proteins, the parameters are customizable. It can be run on the cluster, and anecdotally, it performs better for protein multimers. And so here's just some anecdotes from AlphaFold 3. You can see this person thinks that this is the end of the world as we know it. This transcription factor is very nicely binding into the DNA, this groove in the DNA, so it's very beautiful. But then again, we have people who are skeptical, or it's incapable of predicting this structure. That instead, it forms what they call a ballet tutu. Something something that is not expected for this protein complex. But then again, it's the kind of thing where you just have to try, right? You just have to try different things if you're trying to get a prediction that's accurate. Something else I wanted to mention is AlphaLINK 2. I know more people are using it now than before. So this is including cross-linking mass spectrometry data into the AlphaFold pipeline. If you think about it, when you have cross-linking mass spec, you essentially have relationships between different residues, right? You already know that these maybe should be close in space. And so you can introduce these as weights into the multiple sequence alignment, therefore helping, AlphaFold predict the interaction between proteins. So here is an example where we have a crystal structure and a typical AlphaFold prediction, but when you introduce the AlphaLINK, so when you introduce this cross-linking data, then it will predict it more accurately, right? So it's more accurately reproduces the crystal structure. I should mention this is from the Rep Silver Lab, so they very nicely set this up. At the LMB, we have an implementation that's very simple. You run ColabLINK, your sequence, but now you also give it your crosslinks. I mean, if you're interested in how this should be built, you can just run ColabLINK 2 without any arguments, and you can see what it is. Of course, with all these things, you can always just email me, for example. So yeah, so I know people are using it more and more now, and I'd love to hear after if people have had success with this as well. Okay, let's go through some tips. So one of the main things that people run into when they're running predictions is that their proteins are really big, or they have big complexes, and something that doesn't fit into typical GPUs. I mean, with the AlphaFold server, it's very nice because you can run up to 5,000 residues, but on the notebook that I mentioned, up to 2,000. These are the GPUs that we have right now on the cluster, 2,400, 3,400. You can already do a lot, but for something like Dynein, where you have these enormous complex, there's 4 of them, and then you have this big filament and the cold coil. I mean, just the heavy chain on its own is over 4,000 residues, right? So what you need to do in this case is, I mean, you could try AlphaFold 3, see if you can get it to predict. I mean, with AlphaFold 3, if you put the Dynein heavy chain, it puts it in this really painful conformation where the cold coil is kind of circled around itself. So really, even for big things that you can run on AlphaFold 3, I would recommend that you segment it and run different pieces over in AlphaFold 2 or AlphaFold 3. So here in the heavy chain, I mean, this was built essentially with these different bits and then fit into a cryo-EM density. Even for small things like here's a BICD2 cold coil, you can run this on an AlphaFold 2, but it makes a mistake here where it thinks that there's this long stringy bit between these 2 cold coils. But if you segment it into different bits, you can see that it properly predicts the intermediate cold coils here. And we have experimental evidence for the presence of these cold coils in the center. So I would always recommend. It's also faster and it just better. Mm-hmm. I mean, you would want to cut at the edge of domains typically. You don't want to be exposing bits that would otherwise be buried. That would be my only suggestion. Otherwise, I mean, if you're doing something like this, you can see I've done overlapping regions, right? So you go beyond a bit, and then you start a little bit before, so how if you're stitching these things together, so then you know how to stitch them together. Otherwise, you just have to try. Well, it depends what you're doing. So if you're just making a schematic, let's say, and you want to see what your big protein looks like, then you align to the bits that overlap and use something like Coot or Chimera to stitch them together. You're probably not going to be accurate. I wouldn't then interpret the interaction between different parts of the molecule that come from separate predictions. I would be careful about that, right? But for this kind of thing, I was stitching it however I could, and then using the cryo-EM density to further, relax things into the density. And the combination of those two things give you something that you hopefully is accurate that you can then interpret. Okay, so something to watch out for is false negatives. This is also a truncation thing. So if you have, this long cold coil fragment, here's an example. We work on a lot of cold coils. So here's another BICDR. This is a cold coil, and we have this little peptide that we know binds. We have a structure for it, and yet if you look at this predicted aligned error, I mean, you've already seen it doesn't bind here. But if you look here, there's no confidence in the interactions. And then it turns out when Clinton did this, he just cut it shorter, and you can see all of a sudden it figures out that it is able to bind. Not clear why. Maybe it's distracted by the bits over here, but it is worth trying different fragments of things when you're running predictions. Basically, is the story here. A bit more scary, I would say, is false positives. So this is things that are predicted to be interacting, but shouldn't be interacting. Now, it's hard to know unless you have good experimental evidence for things that shouldn't be interacting. In this case, what I've done is I've cut into this protein and I've exposed something that would otherwise be buried. So I know this would not typically be exposed. And it's happily fit this beta sheet, appended it essentially to this, to the ones that would otherwise be buried here. And, the PAE is quite nice. And, this one could say, oh, look, they're interacting, but I would be careful, right? So it doesn't mean it's wrong, but I would be skeptical about, when the buried residues are exposed, especially. There's also something to watch out for is model-to-model variability. So here we have the 5 different predictions. Turns out this top ranked one is correct. We have a structure for this one. But it turns out that this whole peptide, according to NMR, might interact with this cold coil. And perhaps one would want to investigate, this nicely predicted interaction of this other part of the peptide with this bit of the cold coil. So one needs to be careful that there are other models to look at and not just look at the top ranked one. Sometimes you get lucky and you get something like this where, one of the predictions is folded over and the other one is open. And maybe in this protein, we know that there's an open and closed state for this protein. So maybe one could use this as a starting point for, using other techniques to test whether or not these residues are actually mediating the open and closed conformation. So that was the end of my tips. Does anybody have questions? Yeah, so ranking has been weird. I mean, so initially there was this idea that there's a PLDDT is the right way to rank things. And so the average PLDDT should be the way that you interpret the better result. And then there was a more intelligent way of combining the PLDDT and the PAE, and some percentage of one and percentage of the other is a way of obtaining a number that you can use for ranking. And then there's other more intelligent ways of looking at the PLDDT and PAE, like the local interaction score or the protein interaction score. I mean, these things, I wouldn't treat these numbers as absolute values. They're just an attempt at getting things that are better up to the top of the list. But really, in practice, what you want to be doing is looking at all of the predictions, probably the top few, but I don't have a general recommendation for this is the number to look at. I mean, there's the IPTM, there's a PTM, things like this. Is that okay? I'll talk again about the ranking when I talk about screening. Okay, so applications. So the first obvious thing is, having a prediction helps you build models. I've already talked about this a few times. Here's an example where we have a low resolution model from this work from our lab from 10 years ago now. So Clinton was interested in this bit that he couldn't interpret before. When AlphaFold came out, he was interested in figuring out what's going on in here. So he fragmented this piece of dynactin, this big molecule over here with these cold coil bits. He fragmented out that piece, obtained different predictions, and very nicely could fit those into the structure. And now what you can go do is make mutations in the interfaces, test whether or not, in this case, for example, the autoinhibition happens because of this interaction, or you can use other techniques to interpret this. Even for higher resolution bits, for example, here's a structure of this motor domain where we have these loops. The loops are typically lower resolution, and it's hard to build into these loops sometimes. So it's very useful to run a prediction of this subdomain. You use amber relaxation so that your side chains are a bit better geometry. And then you can stitch this into the problematic region. And at least now you have a first approximation of what this loop should look like. And then you can use your expertise and tools to fit it better. I mentioned construct design earlier. So here's an example where I was trying to purify this protein, BICD2. And I created a truncation that cut through a 4 helical bundle. At the time, I didn't know it was a 4 helical bundle. If you do an N-to-C terminal coloring of this, you see that it actually loops back around and comes this way. But if I had used cold coil prediction software and it told me that the cold coil ended right here, right? And so when I made my truncation, it turns out that probably exposed something that was unhappy, which is why my protein was insoluble. And then now that I had the prediction, I made another truncation that was here, and now the protein is soluble. So this is kind of a nice way of using AlphaFold in your biochemistry pipeline. Something that people have been doing at the LMB a lot now is running screens. So this is basically using AlphaFold to see if you can find new interactors based on a database of either high throughput or low throughput interaction screens, right? So there's now alpha pull down. There's a new version of AlphaFold that came out recently that's very nice. And you can just give it basically either a bunch of pairs of proteins or, everything to everything, and it will run all the predictions and then rank them. And then you can see if you find something new. I've written a version of this called AlphaScreen, which takes UniProt IDs. Something like the BioGrid will give you a list of UniProt IDs. And so you can basically run an AlphaScreen on these. It will generate the FASTA files with little fragments, all the different pairs, and then you can run this on the cluster. And eventually you end up with something like this. And this back to your point, then how do you rank? Now that you have thousands or hundreds or thousands, how do you look at all of them? And so ranking becomes a little bit more important. I mentioned that there's all these different ranking techniques. Again, these are more or less arbitrary. I wouldn't treat these absolute numbers as, fact, but I would just take these as being a way of getting everything to the top of the list. For example, with AlphaScreen, I use this, I like using the local interaction score. I find it brings nice things to the top. But really what you want to do is look through all of them. And more importantly, you want to be looking at the predictions themselves, the structures themselves. And you want to use your expertise about the interaction interface to know if it makes sense. Because sometimes AlphaFold will give you a prediction, but really it doesn't make any biochemical sense. So you really want to be going in and taking a look. There's also this ability to find structurally homologous proteins. So these are proteins that might not have similar sequence, but have a similar structure. And that might be something interesting for your pipeline, for your project that you want to know. We have our very own Kiarash and Joe who have written Progress, which is a very nice, fast way of searching through different structures and finding those that are structurally homologous. We have, there's also FoldSeek and Dolly, if you're interested in doing this, in doing something like this. So here's a Dolly result where we have this protein and we've obtained, a bunch of different candidates for structural homology, if that's something that you're interested in. Okay, so I'm just going to end this section by just talking about some limitations and advanced use cases. So some limitations are, for example, isoform specificity, right? If you have a protein with an isoform, maybe the other isoform shouldn't interact with, this other protein, but it is predicted by AlphaFold to interact. And that's probably because the conservation there, because conservation is used to make the predictions, it's confused by the conservation. So I would be careful with interpreting how different isoforms interact with things. The oligomerization state is also difficult. If you give AlphaFold 1, 2, 3 copies, it'll even make a 3-coiled coil of this protein that we know is a dimer, right? So it's still up in the air how one could interpret the confidence metrics in terms of what oligomerization state this protein should be. So I would be careful then about seeing something like this and saying, well, this protein is a 3-helical coiled coil. Again, we talked about alternate conformations. There's this ability to play with the MSA depth, right? To play with the number of multiple sequence alignment sequences. But there's also a more intelligent way of doing this by using something like AF cluster or ACE. So this is essentially taking your multiple sequence alignment and clustering the sequences using something like a PCA and then running those clusters as AlphaFold predictions. And sometimes things that have the same conformation will cluster together in the MSA. And so if that's something you want to get into, you can check something like AF cluster and maybe to coax some different conformations. Of course, post-translational modifications aren't handled, right? So phosphorylation will just be a gap. And mutations, I would be careful with. I see people making this mistake where they make a mutation in a protein and then they see that it's still predicted to interact. But you should be careful that the surrounding residues are still giving the conservation that the AlphaFold is interpreting as resulting in this interaction, right? So mutations are a bit difficult to interpret with AlphaFold, so I would be careful. Now, RosettaFold, there's a claim that they are able to handle this a bit better. So if that's something you are interested in, I would take a look at RosettaFold. And then with AlphaFold 3 now, we can run ligands, nucleic acids. Before, what we were using is something like AlphaFill or AF2Bind, which would fill in the gaps left by AlphaFold with these ligands. And that's something that you can still attempt if you'd like the AlphaFold 2 result, is to fill in those gaps. But you can also use AlphaFold 3. And RosettaFold all atom is another version of that I mentioned. Antibodies are kind of a special case of this because the way they work is that they rapidly evolve, right? So they don't fit this criteria of having conservation that then AlphaFold can interpret as interactions. So it doesn't work very well with AlphaFold 2 or AlphaFold 3, but there's these dedicated algorithms that one could look at, like IgFold. And like I mentioned, proteins with very low homology. Again, the idea being that multiple sequence alignment is so important, right? they don't work very well. You can try these protein language models, structure predictors that don't require an MSA, but remember that the evolutionary relationships are embedded in those protein language model structure predictors. So they also don't perform very well when there is very low homology. Just something to be aware of, but worth trying. You can also, curate your own multiple sequence alignment. If you know a lot about this protein, you know you can maybe do better than the fetching of the databases. Then I know that this has worked well for some people, is to just make your own MSA, and then there's ways that you can feed that into the AlphaFold pipeline. Okay, we're almost done. Just the last thing I wanted to talk about is structure design. But are there any questions on structure predictions so far? Great. Okay, so really I'm just going to be talking about one, just because it's the one that's easy to use and that we have implemented at the LMB and people have tried, and that is BindCraft. So this is using AlphaFold to go backwards and actually design an interactor based on a target. So here we have a target protein to which I want to design an interactor. And what they figured out how to do is to run essentially this pipeline backwards, where you put a random backbone in, and then you go backwards to the pipeline and figure out what sequence would be an interactor for this backbone. And then you iteratively do this. You keep predicting this, hallucinated interactor with its target until you get something that makes sense. And then what's really cool is that they can put it through this other neural network that makes sure that this binder is soluble, right? Because eventually we want to be able to purify these things. And so it puts it through the solubility, essentially modulating the solubility here, and then putting it through these really rigid metrics of, is the PAE really good? Does it fit kind of the physical criteria that you would want from an interaction? And only if it meets these criteria do we have then a design. And so the reason we're talking about this now is that the claim is that you get very high efficiency, whereas before you would need hundreds and hundreds of binders to try to get one. Their claim is that now you can get interactors, here we have 100% success, nanomolar affinity of this binder to a target, right? And a little bit lower success. Now it remains to be seen if, we can obtain these kinds of successes in our hands, but the appeal is that the input is very simple. There's minimal intervention that goes in, and hopefully you have a high success rate. And so at the LMB, the way to do this is to just run BindCraft with your target. And this target is a JSON file. And essentially you give it, just standard things. Where do you want it? What do you want to call it? What is your target? Which chains in the target do you want to bind against? What residues in that target do you care about binding to? These are the hotspots that you specify. You say, I want to bind on this side of the protein. And what is the range of lengths of your binder that you want? And how many designs do you hope for? And, number of final designs, let's say 100, and then it will stop when it hits 100, for example, in this case. Now, it's not always that easy, but something to keep in mind is you're limited by the cumulative size of your target and binder. So if it's more than if the size of your target binder sum is more than 500, then probably it will not run, or run very slowly on our cluster, for example. So I would keep that very small. And of course, there's a lot more that you can do. A lot of parameters are already optimized and you don't have to worry about, but there's these advanced parameters and these filter parameters that you can play with. So you can basically check out this website, the GitHub for BindCraft, if you want to learn more about these advanced options. So here are some examples. So this is proteins that I work on. We have here, this is my target, and this is my binder. And you can see it's predicted all sorts of different conformations for this binder. It's mostly alpha helical, although that's something you can play with. It's mostly binding on this side. It's been everybody's experience that it likes hydrophobic regions. And in a sense, even if you put in a hotspot, like I told it to bind up here, it has ignored my hotspot because it really likes this side of the protein. Here's another binder that I designed against this cold coil break. You can see it's binding on one side. Really what I wanted here was to bind across the interface, but it was like, no, I want this side. But there's some things that you can do. I mean, truncating down is very important for this kind of thing. If you want to generate many, many binders, that you can then get something that might look reasonable. And if your hotspots are ignored, one thing you can do is mutate the non-hotspot residues. A place that keeps distracting it, you can mutate those to something like a lysine, apparently works well. And then it will start to get out of that distraction. You can also weigh the interchain weights.
And so basically maximize the number of contacts between the chains. Apparently that helps have it look at the hotspot more. And really then it's up to you to purify these things and test the affinities and use all of the things that you've learned throughout the seminars in the biophysics seminar series. Okay. Yeah, I mean, I should mention also that there's RF diffusion. This is another way of designing binders. The reason, I mean, it's not as nicely implemented, so people haven't been using it as much, but it's also very powerful, very nice way of designing binders. Okay, I'm just going to leave you with this list of resources. There's this AlphaFold listserv where I post updates, so you can join this from your dashboard. And I recommend you look at these talks or guidelines because there's very useful information about how to interpret these confidence metrics. Better job than I did today. So I would take a look at these. And thank you. I'll take any questions.